Hi everyone, just wanted to say thanks to ABSS for allowing me to present to all of my supervisors as well. Today I'm going to be discussing one study from my PhD that was looking into neuroplasticity following a complex finger tapping task across two different age groups. As you would know, neuroplasticity relates to the brain's ability to change, both in terms of excitability and inhibitory circuits. Given motor control does decline with advancing age, though, understanding how neuroplasticity impacts these processes may help uncover training strategies to combat declining motor function. However, the assessment of learning in both limbs, or bilateral transfer, and complex finger tapping tasks are underutilized in research, but would help to confirm if changes are task or age related and would assist in formulating future rehabilitation strategies. One previous study did investigate bilateral transfer through a finger tapping task, but found no excitability changes at the thumb. However, they did not assess inhibition. They also found significant performance differences between the two age groups from the finger tapping task as well. But given only the thumb was investigated, it cannot be ruled out that changes in other muscles may have been occurring. And additionally, given inhibition should be assessed, assessing both inhibition and excitability at not just the thumb, but at finger extensors potentially as well, we may be able to further elucidate what the effects of aging are on motor task performance. Therefore, we aim to assess the neuroplastic and motor performance differences across aging after completing a complex finger tapping task. In total, we had 15 healthy young and healthy older 11 healthy older adults participate in the study. TMS was conducted over both hemispheres, both motor cortices, under resting conditions to elicit excitability and inhibitory responses at the abductor pollicis brevis and in the extensor digitorum communis, so muscle of the thumb and digit extensors. Following the pre-training TMS measures, participants were familiarized with the task itself, the movement sequence, and then their performance was assessed on both their left and right hands, with performance being the number of correct sequences completed within a 30-second period. As indicated in the video, then participants completed the task training on their dominant hand only, which involved the same movement sequence, but at a set pace for 5 lots of 1-minute rounds. The same outcome measures were then taken post-motor task training. Data was assessed for normality, and as TMS was not normally distributed, it was converted to a pre-post change score and assessed by an ANOVA with group and hand as the variables of interest. Behavioral data was normally distributed, so an ANOVA was run with time, hand, and group as the variables. We also used the behavioral data to generate a bilateral transfer score for each of the two groups, and where appropriate, post hoc t-tests were undertaken. For the behavioral data, we did not find a significant group effect or interaction, but what we did see was a significant time effect and time by hand interaction. Post hoc analyses revealed that all the post measures, whether it was the trained or untrained hand, were significantly different to the pre-training measures, indicating that there was an improvement in performance and that bilateral transfer had occurred. Again, this was without separating the two groups, as we didn't have that significant group effect from the ANOVA. And in addition, there was no between group difference on the bilateral transfer score. So together, this demonstrates that bilateral transfer is not different between groups and therefore seems to be maintained with advancing age. This is in contrast to Dickens et al. That I spoke about earlier, who did show statistically significant differences in performance between their older and younger groups. Potential reasons for this difference is that the sample size in the current study was a little smaller than theirs, or the age discrepancies as well, with my older adult group being slightly younger than theirs. Moving to the TMS data, there were no significant differences at the abductor pollicis brevis muscle between the younger and older adults, which supports Dickens et al. By showing no excitability differences and adds to their results because we additionally showed there weren't any inhibitory changes either. For the digit extensor muscle, which the graphs on the screen represent, we observed significant decreased inhibition as shown by an increased Sicky ratio for the older adults. This difference indicates that the extensor digitorum communis may be more relevant to this motor task compared to the thumb muscle itself. But what might this difference mean? The fact that training caused alterations in the untrained hemisphere in older adults may be evidence of mirror activity, whereby there's unintended movements occurring on the opposite hand to that which is being trained. And this is more common with advancing age as well. If this difference in the Sicky that we're observing is evidence of mirror activity, then we would likely expect to see a difference between the hemispheres of the younger adults that is not present in the older adults. When investigating this, that is what we saw. The younger adults here have increased inhibition represented by the decreased Sicky ratio of the untrained hemisphere as a result of dominant limb training. However, the older adults do not present with any such hemispheric lateralization. This phenomenon of reduced hemispheric lateralization is termed Harold or hemispheric asymmetry reduction in older adults. This reduced lateralization could be evidence of de-differentiation, which represents a breakdown in the appropriate selection of brain regions associated with task performance, while others argue it is representative of a compensatory mechanism to account for age-related declines and enabling older adults to maintain task performance. Based on our current results, though, we can't specify which is occurring here. Overall, while our study has some limitations, including the small sample size, our results indicate bilateral transfer is maintained with advancing age. Additionally, we indicate the importance of the extensor digitorum communis to the complex finger tapping task over just the thumb muscle itself. And we have showed specific age-related inhibition differences that weren't previously observed in this task as well. To further explore neuroplasticity, motor control, and aging, research should aim to disseminate if the findings, the age-dependent changes that we're seeing here are, one, observed in other complex motor control tasks, and two, if they are evidence of compensation or de-differentiation. Thank you.